Hello, welcome to our lecture number 6 on single-cell high-throughput data science. So today we are going to talk about the feature selection and the identification of highly variable genes. But before we go to the concept of the feature selection, I will continue to mention a topic which we have some leftover from the last lecture. So last time we mentioned about how to do matrix multiplication operation. And that's a definition. So once you have this 2 by 3 matrix and you want to multiply it by another matrix which has to be 3 by 2, and this you have to have a match between these dimensions. And the first matrix, the second dimension has to be same as the first dimension of the second matrix. Otherwise, you just cannot do this multiplication between two matrices. The way to do it is put them into this position, and then the outcome of this product will be a 2 by 2 matrix of this. And we also mentioned what's the meaning of this multiplication. I gave you an example of using a hypothetical scenario which you and your roommate want to make sandwich, and then you have 3 kinds of sandwich, and they have a different ingredients. So you have a bread, a peanut butter, jam, etc., etc. And then each of you want to make first type of the sandwich. You have 4 and your roommate have 3, and then for the second type of sandwich you have 5 and your roommate want to make 3. At the end you can compute how many each of the ingredients you need in order to make your set of sandwich and your roommate's set of sandwiches. And this 2 by 3 matrix and multiplied by 3 by 6 matrix will end up with this final production, which is a 2 by 6 matrix. So this example will give you some idea what we are doing here, why we are trying to multiply by with one matrix against another. Okay, the next concept I want to make sure you understand is we want to use vector to represent your data point. Okay, vector is we can think about it as a n by 1 matrix. Okay, because this second dimension is only 1, so it's a vector of the data. And this is your data point, and that will be the variable or characteristics to describe your data point. Okay, if you have a data point in 2 dimensions like this position A, then you only need 2 parameters to describe the position of this A. And we always consider this as origin, and then you have x-axis the value and the y-axis value. So this data point A can be defined by the value of x and y. So this is a 2-dimensional space scenario. Your data point can be written in the vector form, which is the y-x and the y. So that's your data point A. Okay, similarly, you can have another data point B. This will be another set of data for B, and Y for B to describe the position of this data point here. So you have A and you have B, and so the idea here is the data point is on a 2-dimensional space. So you need 2 variables to describe it. And in this scenario, you have n dimension of this space. You have a vector of n numbers, to describe the position of this. Okay, in this example, n equals 2. This is the same situation as we have shown here. Okay, you have 2 values to describe a data point on the 2-dimensional space. Well, when you have n equals 3, you can have x and y and z, 3 numbers to describe this position of this data point. If you have a higher dimension, in this case n equals 15, and then you will have a series of numbers, 15 of them, to describe your data point x. Okay, think about this as a cell in our single-cell RNA-seq study, and then you will have n numbers of gene to describe the position of this cell in the high-dimensional space. So that's a geometric interpretation of the vector. So next slide, I'm going to show you an example of matrix and vector multiplication. So that's the key to understand how a matrix can be used as a transformation or as operator. Okay, operator. So now we will have a completed new understanding of what a matrix is. In the previous slide, we showed a matrix will describe how different gradients will be composed into a sandwich. But in this case, A will not be a component. Itself is operator. So you can use this to transform a data point. Okay, so let's see, you know how to do the multiplication between 2 matrices. So if you think this vector as a special case of the multiplication, one of the dimension is 1. So you can do multiplication for A multiplied by X. And then you will obtain the answer of this. And after the multiplication of these 2, you get a new vector 3, 6. So if you think again, and the product of this is a new data point. Okay, so the original X, the position, the 1, 1. So that's the X in this case here. And after multiplication, then you have a new position for this transformed data point. It's in the 3, 6. So the new position, the data was here. And then after transformation, let me change the color. And you got a new data point at 3, 6. Okay, so what's the transformation of matrix A when you apply this to your original data X and change the X to become AX, and the AX becomes longer. Okay, the distance from this to the origin is longer, and then also the angle changed. So you basically rotate the data point from this position to another direction from the origin. So the effects of this transformation by matrix A applied to data point X is to stretch, so scale the length of to to the origin, stretch the data point, and also rotate. So in that case, matrix A is a transformation which rotates the data point and also stretch its distance to the origin. Okay, so that's what the A means. That's what I want you to remember. When you use matrix A as operator, you actually can use it as an operator or transformation operation to apply to data point in the vector. Another I want to pay attention that this vector is a little bit special. So this is another data point. So if you apply the same matrix A as an operator to this vector, this data point, and you get the output which this, and the special about this vector is after you multiply it by matrix A, it only change the scale, not the direction. So it was in the 1, 2 position, 1, 2 position. And after transformation, you will still point it to the same direction. But the only difference is you stretch it by 5 times. So it's 5, 10. It was 1, 2. Okay. So, so this is a very special vector, a very special data point regarding to what this matrix can do for it. So if you have if we write an equation, then you have a multiplied by this special x is equal to and 5 times of this special matrix vector. Okay, so that's in general form. If you think this is a constant number, so you can replace this relationship writing as x equals lambda x. Okay, so this is very special vector, which in our case is 1, 2. So this special vector is called eigenvector. Eigen is a German word, means characteristics, eigenvector. So this eigenvector is a special vector regarding to the operator matrix A. And this lambda, which in our case equals 5, is eigenvalues. It's eigenvalue. Okay. Eigenvalue. So eigenvalue and this eigenvector is a set of special combinations of vector and the constant to reflect the property of A matrix as an operator. All right. So the eigenvector and the eigenvalue is very important concept in lineage algebra and in machine learning. So basically, you can use this eigenvector and eigenvalue to describe the property of matrix, a squared matrix A, as a transformer, as an operator that can transform other data points. All right, there are a lot of applications with this, but for this lecture, I just touch on base of these 2 important concepts to give you a quick idea about this. So next, I'm going to continue to mention the clustering algorithm, which is a special form of the clustering called spectral clustering algorithm. Remember, this is a advanced technology you can apply to the scenario like this very simple condition. You have 3 clean groups, and we know k-means will do the very good job for this. So spectral clustering will be very good as well. And in this scenario, if you have 3 clusters and they are forming this very special formation, and the k-means algorithm will not do a very good job. However, spectrum clustering will also apply here and will recover this separation of the clusters as you expected. Remember last time we say k-means will not do very good job for non-convex shape of this. So if you have data points from this shape, will not be k-means algorithm will not be able to do a very good job to classify them into a separate group. However, spectral clustering will recover this relationship. So I quickly move into some details of how spectrum clustering will works. So first, we will describe your data points. So here we have 6 data points. We use a relationship between them by link them by their distance, probably form a network. So this network can be represented by a matrix called adjacency matrix. Okay, so if you have 6 nodes, in our application is 6 different cells, and by their profile, by their distance, you can link them. If their distance is small enough, you will make a link between them. And once you have this network, you can represent it in this adjacent matrix. This is a squared matrix, and by n, if you have 6 nodes, then this will be 6 by 6 matrix. And if 2 of nodes are linked, so in our case, 1 and 2 are linked, then you put 1 in this position, 1, 2 position. And the 2 and 3 linked, and then 2, 3 will put a link, 1, 3 linked, 1 here, and so on and so forth. And also this network has no direction, so 1, 2, and 2, 1 will be they're always linked, so by either direction. So this matrix is a symmetric. So once you have this matrix, you can use this matrix to replace the real network, because all the information has been already captured in this matrix A. Then you will compute a degree. Degree is how many partners of each node are linked. So for node 1, you have 1, 2, 3 neighbors linked, and then the degree for A is 3, and then for 2 also there are 2 partners are linked. So you fill in this diagonal position of this matrix and put all the others as 0, and this matrix is called a degree matrix D. It's very easy to compute. All you have to do is to summarize all the columns of the adjacent matrix. For example, this is 3, this 2, this 3, this 3, this 3, this 2, and then you put this degree. It's called a degree, and put into the diagonal position matrix. Then you have a degree matrix. So once you have that, then we compute called the Laplacian matrix. Okay, Laplacian matrix is also very easy to compute. It's also a symmetric n by n matrix. The Laplacian matrix is the difference between the degree matrix and adjacency matrix. Basically, you use a distance matrix degree matrix minus the adjacent matrix, then you obtain this so-called Laplacian matrix. And because it's minus here, that's the negative values in the off-diagonal position. So once you have this, that's the key. We are going to do eigenvalue, eigenvector computation, decomposition of for this squared matrix. So that's the main steps for the spectrum clustering. You have the data points, make a matrix, and you compute the Laplacian matrix. Then you will compute its eigenvalue and eigenvector, because you have multiple eigenvector solutions, and it forms matrix. And then so this found eigenvectors and eigenvalues. Then you will find the second smallest eigenvalue and its corresponding eigenvector, and use this to separate the data points into subgroups. Okay, in this case, we select this second smallest eigenvalue corresponding eigenvector, and use this position, like first three are positive, last three are negative. So now we know we can separate this into two clusters, and left and right by their sign. So this is a basic idea how you perform spectrum clustering. Okay, so now I quickly finish off the introduction of clustering algorithm. I will jump into today's lecture, and before that, let's go back to our workflow of a single-cell RNA-seq analysis. Remember, we have raw data, UMI matrix, which is a gene expression level for single-cell gene and cross cells. And then we have a dimension reduction algorithm using tSNE or other algorithm to visualize or reduce the dimension, and then use clustering algorithm to separate the cells into groups. Then you can assign the cell types by looking at a particular gene, so-called marker genes expression across specific cluster cells. Okay, today we are going to introduce one step before you do this dimension reduction. This step is called feature selection. Okay, so let's look at this workflow. And you have an expression matrix, and then before you do this dimensionality reduction, you can do a feature selection to get rid of some of the cells. In this case, those cells removed. You can also get rid of some genes to reduce the dimension. Okay. So we will emphasize how to remove some of these less informative genes. The purpose of this feature selection is try to increase the efficiency, computational efficiency of the downstream process of data, and use less computational power, and get rid of some noise and less important information from the original matrix. So in order to get some performance for downstream analysis. So that's the purpose of this. Again, I think this step is optional. If you have a relatively small data sets, you don't have to do it, but it will become more and more important once we have a larger and larger matrix as the input. So from definition, feature selection is a process where you automatically or manually select those features. In our case, is for cells. Then those features will be their genes, which gene you want to include in the downstream analysis. So these features will be selected based on their contribution to your prediction variables or output in which you are interested in. So we want to keep those most important genes in order to gain some performance for downstream. Irrelevant genes, features will be decreased. Will be when you use the decreased accuracy of a lot of models. Okay, so that's a concept of feature selection in machine learning area. Okay, back to our single cell data analysis. Feature selection is to select the genes that contain relevant biological signal. Because remove those genes have very little contribution to any subsequent analysis. There are example applications. Okay. Then how to do it, that's what we are going to talk about. So there's a concept called highly variable genes. It's what we are going to extract from the original expression matrix. Highly variable genes based on assumption that gene with a high variance relatively to their mean expression are due to biological effects rather than just technical noise. And this method seeks to identify genes that have a higher variability than expected by considering relationship between variance and the mean expression. Okay, so there are different ways to do it. That's the concept we are going to introduce. So in 2018, and this is a Hong Kong University group, conducted a study to compare different methods that have been designed to identify the highly variable genes. So there are 7 different highly variable gene identification methods, and they compared them and found their overlap between their results are not so high. So only a few genes can be identified by all the methods and regarding them as a highly variable genes. So it's still an active statistic problem, and the people still trying to find the best method. Okay, so there the statistic basis for this is we needed to find a measure for the variation or variability to describe the dispersion of gene expression across cells. There are many different measures we can use, the variance, standard deviation, so they all valid statistics measures, metrics that you can use to describe the variability. CV is one of the commonly used, so it stands for the coefficient of variation, and that's the ratio between standard deviation and the mean. Okay, so if you have a vector of data you want to compute, say for example you have a gene in the not a gene, a cell, then have an expression of a different gene So I'm wrong. So you have this is a gene, and then cross cell you have a different value of this gene's expression, and you can compute standard deviation for this gene across all the cells. Then you compute the average gene expression, the ratio between these two, give you the CV of this gene expression across cells. Okay. So that's the definition of this CV, and the standard deviation is the square root of the variation. So you can express this if you have a variation is the standard deviation. In assumption of the Poisson distribution, Poisson distribution have a property that the variation and the mean are the same. So we can replace, if you assume your distribution of the data is follow the Poisson distribution, then we can replace the variation with the mean. In that case, this CV can be rewrite as this form. So there's only mean is related, is relevant. If we take a log across both sides, and then you will have this negative 0.5 log mu and log CV. And this is a constant value. So the whole log value of these two have a linear relationship. Okay, so that's what the relationship we are going to use to capture the assumption which your these two variables are linearly related across all the genes. So this applies to technical variability, and technical variability gave you the data which follow the Poisson distribution. So let's put them together. Okay, so this is, looks very busy, and each point here is a gene. Okay, so this is gene 1, this is gene 2. For example, that's all genes. Then we can compute this gene's expression across cells. And we found the gene 2 have an average expression in this level. And then we can compute the CV for this gene, and it's here. So for this gene, it's have a mean of this level, and the CV at that level. We do this for all genes, and then we see a clear negative correlation between genes mean and genes CV among cells. When you consider all the genes, all these blue points. And when you do the regression between these two variables, as we described, this log-log relationship is negative and linear. So that's where you get this right theoretical expectation if you assume your data points follow the Poisson distribution. Okay, so because the Poisson distribution, their mean and the CV in this log. So look at the x and the y axis in the log scale, and then you expect to see this negative linear relationship. And you can also fit your real data. So these blue points are from real data, is not hypothetic. If it's hypothetic and follow Poisson distribution, every blue data point will be on this red regression curve, regression line. However, now you can follow the real data and do the regression. And here for highly expressed gene, they are not follow the Poisson assumption very closely to this red curve. So that's why we need a nonlinear curve to follow the pattern. So we describe this because we want to capture some of those non-biological related variation among genes. And after that, we compute for each gene their observed CV. So for example, for this gene I, we have its observed CV with compute based on data is here. However, according to its regression line, its green line, it should have a expected CV is here. So that's observed CV and this expected CV based on the model we have built for the same mean. So this mean, if you have a gene expressed at this level, you expect to have this level of CV. However, in reality, you obtained a much higher CV. Means for this gene, across cells, this gene's expression is more dispersed than you can model use Poisson distribution. So we are going to compute the difference between observed and expected. So this difference is we call residual CV. And this CV can be used to describe the true CV we observed. We use this to measure, we use residual CV to measure this gene's expression variability across individual cell. So that's the idea proposed in 2013 by Brandeck in the paper, and then here they use CV squared, the same idea. They use the residual CV squared to found genes which have a higher variability in their single cell expression compared to expected value. And those genes will be identified as a highly variable gene because their expression variability measured by CV is higher than expectation, much higher. So this is the method we are going to use to identify the highly variable genes. Over the year, there another methodology has been proposed. There similar idea. Okay, it's log CV average. It's very similar idea and slightly different fitting regression algorithm applied here. There another way to do it. This is not a very well accepted method, so I'm not going to mention into any detail. There are other models you can use for the feature selection for identify the highly variable genes. Have been okay. Let's move on. So far, I mentioned that single cell RNA-seq provide our ability to identify highly variable genes, and highly variable genes identification and it's a main approach to do the feature selection. You want to use those highly variably expressed gene as a much smaller data set to do downstream analysis. The reason is you can use those highly variable gene to capture the diversity of gene expression across cells. For example, in a traditional scRNA-seq, you get per sample just one gene's expression for all the genes, but each gene has only one data point, so there's no way you can compute the variability about the gene across samples. However, for single-cell RNA-seq data, you have each gene, you have multiple expression measured in across many different cells. That's why you can compute the CV or mean to measure the variability across all the genes. And those genes have a highly variable, high variability is important, like something like this gene is important, gene I, because these genes are highly expressed in some of the cells, but not the others. It's highly expressed in those cells and lowly expressed in those. So this gene is biologically valuable to help you to identify the different cell types. So if you think based on this marker gene, and those are the one group of cells, and then the rest of them is another group of cells. So this column is more important biologically compared to this column. For example, there are no gene expression at all. So feature selection will help you to select those kind of genes and delete some of those less informative genes to reduce the dimension and use these highly meaningful genes to do the downstream analysis. So that's one purpose of this feature selection of highly variable genes, because they tell you the difference between cells. Okay, next I'm going to present a usage of highly variable gene selection measurement method, but applied to opposite purpose, which we are going to use highly variable gene to measure gene expression variability among a group of cells from the same cell type. So something like this, they are highly similar cells. There's no cell type difference, and also they're in the same shape and they're synchronized. So still, if you use single-cell RNA-seq analysis across those cells, we will find a lot of gene expression noise. So the noise is also the description of this single-cell gene expression variability, but because all these cells are from the same cell type, so those variability we can see across cells described as a noise. Okay, so this is an example of select the genes across those gene cells and to have this different expression. And you see some gene expressed at a high level overall, but all those genes have a variability across cells. And Kim Jung-yoon, a professor from Pennsylvania University in 2015, proposed the idea that those variability or variation is important for cell function. So he proposed the idea called variation is a function and asked a hypothetical question: are single cell differences functionally important? So at that time, we cannot test this hypothesis because single cell data is still limited, and so we don't have enough information. we can formalize his hypothesis as whether gene expression noise is related or is a function itself. So we go ahead to sequence and collect the data from 3 different cell types. One is from a lymphoblast cell line, and then another from a long airway epithelium cell, and then collect data from single cell from a dermal fibroblast, and another more. And then using LCL as example, I'm going to tell you the filtering process in order to collect a highly homogeneous population of cell. So even for this cell line sample, we know there are cells in G1 phase, S phase, and G2 phase, so we can use marker genes to identify those cells. And then we get rid of the other cell phase and only keep cells in G1 phase as our input cell type, cell states. And then we remove mitochondrial genes and normalize library size, and also use dimension reduction to see the cell relationship and select a group of homogeneous cell population. So we did all this step, and then that's visualization, and we know PHATE can show the structure among cells. So you see, for the same set of cell and the representation of the dimension embedding the different, and this gives you more separation of the data points. So we use PHATE as our visualization tool, and then select a group of cells in the center of this group. So assume all the cells in there, by the way, they're all G1 phase cell from the same cell line. They should be a highly homogeneous population of cell, and we keep those cells in the downstream analysis, and then you use tSNE to visualize this no matter what parameter you use, and you always have a cloud of cells very similar to each other, so confirm our selection of the cell. So among this group of homogeneous population of cell, we compare their gene expression variability. For example, these 2 genes as an example, and this is a small unit of ribosome protein, and this is an immunoglobulin gene. And across the cell, you see these 2 genes expression have a different pattern. And when we plot them out, we see, okay, this gene is homogeneous population of cell. In this homogeneous population of cell, uniformly expressed across all cells, but in a relatively low value, but globally as a across the whole population, expression level is higher enough. And for this immunoglobulin heavy chain constant gene, we see a very different pattern. A lot of these cells do not express the gene at all. So you have a 0 expression level, but once this gene is expressed in some of those cells, the expression level is really high. So here is 600, same scale as this panel. Although on average, so the mean expression of these 2 genes are the same, very similar to each other. However, the variability is very different. So we use CV to measure this. Remember, CV is the standard deviation divided by mean. These 2 genes have a similar mean across cells. However, the CV is much higher for immunoglobulin gene in the 6.38, and this was only 0.43. So we can use this method we presented before, that is for selection of highly variable gene, and you plot out the relationship between the mean and the CV. Here we use C squared, and in the log plot, and it's a relatively linear negative correlation between these two parameters. And then you can do this regression to predict the pattern across all genes, and any gene which above this curve have relatively high variability, such as this one, and that's our ribosome protein examples, which under this curve is more uniformly expressed. Those genes are these non-highly variable genes, and those genes are highly variable genes on the top. So here a comparison between these two genes, which is here and here, against the 4 genes which have a highly expressed, highly variable genes on the top. You see the pattern difference. This is the scale for these genes is 0 to 35. This is 0 to 10. Here is 0 to 1,000. Here's 0 to 200. So a magnitude of the largest expression to the lowest is much large in higher variable expressed gene. Will those non-highly variable genes expressed more uniformly across cells? Okay, so that's the difference between them. Now our question is, once we identified those highly variable genes in the cell line, whether those highly variable genes associate or enrich with any particular function. So for lymphoblastoid cell lines, we identified several these highly variable genes, and then we studied the enriched gene ontology term associated with these genes, and those are the results. Clearly, the immune system related gene sets, including cytokine-mediated signaling pathway, defense response, responses to chemicals, and the enriched pathway tell you the same thing. And we know lymphoblastoid cell lines derived from the B cell, so it makes sense those functions are associated and consistent with where the cell lines are derived. So this tells you that highly variable genes really carry the essential function which reflect where the basic function of the cells, okay, in which the highly variable gene was identified. Remember, we tested several other cell lines. So this is a long airway epithelium cell, and then those are the highly variable genes identified in this cell line, and you will see those gene sets are different from what we identified from the lymphoblastoid cell line. So they cross different cell types, they don't share any of these highly variable genes. And this gene set enriched with the function, for example, as a collagen formation as a representative function. So again, this will tell you the function associated, which is linked to this particular cell type. Cytokine is also, so these cell lines also have some immune system related functions. So those there are some overlap with the lymphoblastoid cell line, but overall, this highly variable gene will also reflect what the function of the cell type is in which this highly variable gene is identified from. This is gene enrichment analysis for dermofibroblasts. Again, there's a different set of highly variable gene, and that's a function enriched and associated with this set of genes. And we see a key term such as keratinization, which is a specified for this dermofibroblast, and also directly reflect the function of those cell type. So taking them together, we can see from using highly variable gene, you can identify the most important feature function which associated with the cell type in which the highly variable gene is identified. Then, as I mentioned, we test another cell type. In this particular example, it's different from all the other 3 cell types we identified. For all these 3 cell types, we found a lot of highly variable genes, which those genes clearly above or deviate from the regression curve. However, this cell line has very little or no highly variable gene can be identified. So this particular cell line is induced pluripotent stem cells, so iPSCs. So this gave some idea that looks like those cell lines have no. This cell line has no highly variable gene, identified, and then accordingly, there's no enriched function associated with this cell line. So this gives our idea the difference between these 3 and this stem cell is this is undifferentiated. And all these are the highly differentiated cells. So what we can tell from this pattern is when you have a differentiated cell type, then you expect to recover a set of highly variable genes from them. In contrast, for undifferentiated cells, such as iPSC, those cells have not been registered to any particular biological function function yet. I mean, differentiated cell function yet. So there's no highly variable genes. Okay, so we can see using the feature selection function to identify highly variable genes is not only helping to reduce the dimension and reduce the burden of computation, but also measure those variability across genes. You can use this as a research tool to capture the function of the differentiated cells. So if this hypothesis, if this result is correct, we expect if you have a cell line which in the middle of iPSC and derived into different cell types, but not yet complete, you will see an intermediate level of this feature of highly variable gene. So in iPSC, there's no highly variable gene, but in this stage, you start to see some of these highly variable genes being created, but not as many as those highly differentiated cells. So this cell is neuron stem cell, and it's in the middle of differentiation from iPSC to more matured neuron or glial cells. So this cell type is intermediate step of this differentiation, and we start to see the increase, the pattern of the highly variable genes start to show off. So this comes from what we have just discovered, the difference between undifferentiated and differentiated cell types have a different levels of a highly variable gene can be detected. So in conclusion, we can use highly variable gene detection as a research tool to define the function of unknown cell types. Okay, thank you for your attention. That's all for this lecture, and see you next time.